In this lecture, we will be able to discuss the physiological effects of auxins. So, first physiological effect that auxin cause is cell elongation and longitudinal growth of plant. Auxins are well known to promote elongation of stem and coleoptile. However, this effect is not observed when the treatment of exogenous auxins is there or when we externally apply the auxins to the intact plant. We will not be able to see its effect. Probably it is because the required amount of auxin is already present in the plant. However, it is observed that exogenous application of auxin promotes the growth of the apex if we remove the apex of the shoot before the auxin treatment. This clearly indicates that the growing apex having the meristematic cells is the site where endogenous auxins are present in sufficient amount. Once the apex is removed, the source of auxin is also removed. Therefore, decapitated stem and coleoptile which do not have endogenous source of auxin, they respond to the externally applied auxin. Extensive studies on the mechanism of action of auxin in cellular elongation show that auxins promote the cell elongation by causing increased plasticity of the wall with decreased plasticity, by enhancing the uptake of water and solute, and by interacting at the gene level by synthesizing enzymes that are required for the synthesis of cell wall components. Auxins induce enzymatic or non-enzymatic deformation or loosening of the cell wall by breaking the cross links between the cell wall components, which results in increased plasticity with decreased elasticity of the cell wall. Next physiological effect which we commonly see in nature is apical dominance. So it is a common observation in vascular plants that, especially in the taller ones, that if there is the apical bud present in the intact plant and it is growing under natural conditions, the lateral buds remain suppressed. Removal of apical bud causes fast growth of the lateral buds. The influence of apical bud in suppressing the growth of lateral buds is known as the apical dominance phenomenon. According to Thiman and Skoog in 1934, the auxin synthesized in apical meristem portion is translocated downwards and inhibits the growth of lateral buds. During the downward movement of auxin, some correlative inhibitors are synthesized which inhibit the growth of lateral buds. However, if we remove the apical bud, the influence of auxin is destroyed and the growth of lateral bud occurs very rapidly. Next physiological effect is root growth and root initiation. Auxins promote root growth only at extremely low concentrations, that is a concentration of 10 raised to power minus 7 to 10 raised to power minus 13 molar concentration. However, at high concentrations, auxins inhibit the root growth. In fact, the concentration of auxin which is most suitable for shoot elongation is always inhibitory to root growth. This inhibition is thought to be caused by the production of ethylene. The concentration of auxin, which is inhibitory to root growth, causes initiation of adventitious roots from the nodes or basal portions of the stem. For this reason, the auxins, particularly IBA, that is indole butyric acid, and NAA, that is alpha and beta naphthalene acetic acid, are used by the plant growers to induce root formation in stem cuttings and cut leaves. These synthetic kind of auxins are more effective in the initiation of new roots. Probably it is because they are not easily destroyed by the enzyme IAA oxidase, and therefore they can persist longer in the plant. Next physiological effect which we observe is the prevention of abscission layer in the plant. So, for understand this phenomena of prevention of abscission layer, we must understand the phenomena of abscission. So, what the abscission basically is, it is the shedding of mature leaves from the stem or the ripened fruits from the stem. Generally, a layer of tissue is formed at the base of the organ. For example, a layer of tissue is formed at the base of petiole in mature leaves in case of deciduous plants. This layer of tissue is called abscission zone. The cells of this zone are thin-walled and without deposition of lignin or suberin. At the time of abscission, some phenomena occur within the plant, like the middle lamella may dissolve between the cells of the two middle layers. Middle lamella and primary walls of the cell of middle layers, or the whole cells of middle layer may dissolve and cause separation of plant organ. It has been shown that abscission zone doesn't occur when the concentration of auxin is high in the plant, particularly when the gradient of auxin is steep, that is, more auxin is present on the distal side and less auxin is present on the proximal side. The abscission zone formation occurs very rapidly when this gradient of auxin it becomes less or neutral, that is, there is less concentration gradient of auxin. Moreover, the plant hormone ethylene is found to promote the layer of abscission formation. Thus, a high concentration of auxin prevents the formation of abscission layer, and the phenomena is controlled by the concentration of auxin and ethylene. Next, another important phenomena of or physiological effect is the effect of parthenocarpy. Auxin is well known to induce the phenomena of parthenocarpy in the plants. So, the phenomena of parthenocarpy is the formation of seedless fruits without the process of fertilization in a number of plants. External application of auxin on flowers causes the development of seedless fruits in many of the plants, such as tomatoes, such as apples, cucumbers, etc. Respiration. A close correlation has been observed between the auxin-induced growth and increase in respiration in many of the plants. The effect is thought to be related with auxin-induced synthesis of various enzymes, which results in an increase in the process of respiration. Another phenomena which has been observed, or another physiological effect is the callus formation. Initiation and promotion of cell division by auxins is very useful in tissue culture experiments and formation of callus. Callus is basically an undifferentiated mass of cells. Healthy growth of callus depends upon the auxin which has been added into the culture. Now, the vascular differentiation. Auxin is responsible for initiation and promotion of cell division in the cambium. This effect of auxin is particularly important in secondary growth of the stem and differentiation of vascular tissues, that is xylem and phloem in the plant. Auxins are also an important herbicides. Many synthetic auxins such as 2,4-D, that is 2,4-dichlorophenoxyacetic acid. Another synthetic auxin is that is 2,4,5-T, that is 2,4,5-trichlorophenoxyacetic acid, etc. They are used as selective herbicides. Herbicides are the chemicals which eradicate the weeds in the field. They are widely used as potent defoliants due to their destructive properties. There are many instances where these chemicals are used in the chemical warfares due to their destructive properties. For example, Agent Orange, which is a mixture of 2,4-D and N-butyl ester of 2,4,5-T, was used as a defoliant in the war. Thanks.